Hey guys, so today we're going to go over part two of protein synthesis, and this is going to go ahead and take a look at translation. I'm also going to take some time to go ahead and tie back in proteins, which we went ahead and talked about back when we were looking at our macromolecules. So let's go ahead and take a look at our essential questions for today. First of all, where does translation take place? So much like when we were talking about uh, transcription, we got to make sure we understand where translation takes place. Number two, what is the role of all three types of RNA? So if you did the activities from last class, you should know that there are three primary types of RNA. Where are their primary functions carried out and what do they do? And third, what must happen following the production of the polypeptide in order for us to have our final protein? All right, so quick review from last class. We have this process of protein synthesis. This is the way that we make the proteins. And remember, we've talked a lot now about how proteins do all the heavy lifting in your body. DNA is important because it's the instructions, but it doesn't actually do anything. It's the instructions to build proteins which do almost everything. There are two parts to the protein synthesis process. The first we went over last class is transcription, and the second is translation. Now, we focused on transcription last class, so this class will be really taking a look at transcription. Here's our overview of protein synthesis. Notice that we've identified two key RNA molecules that we introduced you to. We have the mRNA or messenger RNA, which is there to go ahead and take the message from DNA out to the ribosomes. And remember, it's there because we don't want the DNA to leave the nucleus so that the DNA, the original blueprint, stays protected within an extra layer of security, the nuclear membrane. It's going to head out to the ribosome, which is where translation that we're going to talk about today takes place. And in translation, mRNA is going to match up with another RNA molecule known as tRNA or transfer RNA, which is going to convert the message from RNA into the amino acids that are going ahead and build our protein. All right. So, transcription. First steps. Remember that transcription takes place in the nucleus. It has to because the nucleus is where the DNA is stored, and we don't want the DNA to leave that storage area so that it can stay safe and protected to minimize the number of mutations. First thing we have to do is to unwind and unzip the DNA double helix because otherwise we won't be able to actually get to the nucleotides, specifically the bases that we need to go ahead and copy to go ahead and make our mRNA. Now, unzipping, remember what we're talking about here is breaking the hydrogen bonds that hold those nucleotide bases together. When we're doing transcription, the key protein involved is RNA polymerase, which is going to go ahead and copy the DNA into the initial RNA transcript, creating codon triplets. Remember, everything's read in groups of three. DNA triplets are simply called triplets, but mRNA triplets are called codons. That's where your codon wheel comes in. The initial mRNA transcript is then going to be edited. We're going to keep the exons, remember exon for expressed, and remove the introns for intervening sequences. Then we're going to go ahead and send the messenger RNA out of the nucleus through a nuclear pore, as you see here in our demonstration, where it's going to go to a ribosome, and that ribosome is going to pair tRNA with mRNA to go ahead and produce our amino acid sequence. All right, so this time we're going to focus on translation. The cell's going to use the messenger RNA to produce proteins. Remember, DNA is found in the nucleus and proteins are made at the ribosomes. So in this particular image, A represents the nucleus. You can go ahead and see the mRNA leaving the nuclear pore and heading towards the ribosome. C then is going to be the ribosome. And at this point, you're going to see that at C, the mRNA, which is G, is going to match up with a tRNA represented by D. At the top of D, you see that little circle, which represents the amino acid that is being brought in for that particular part of translation. Okay, So some key concepts in translation. Remember, this takes place at the ribosome. There are two parts to the typical ribosome, the large and small subunits. They actually come together for translation, and they're going to go ahead and grip in on the mRNA molecule. If you ever used one of those label makers that were really popular in the late 80s, early 90s, where you kind of click the trigger and it moves along, this is very much how the ribosome works in terms of making amino acid sequences out of the mRNA. It's going to read triplets from the mRNA, which remember are called codons, and it's going to begin the copying process at what we call an initiator or start codon. It's going to continue reading in triplets, codons, until we hit a stop codon, where it's going to terminate the chain. When it's reading these, it's matching them up with a tRNA triplet, which is going to be called an anticodon. And that's going to go ahead and make the chain longer, creating peptide bonds, which are the bonds that are going to exist between the amino acids, until it gets to the stop codon where chain termination occurs. So let's take a look really quickly at the tRNA molecule. Now, the typical representation that you go ahead and get in a lot of biology textbooks is what you see here on the left. At the very top, you have your circle, and this circle is going to represent right here. This is going to represent the amino acid, in this case, leucine. Then you've got your hydrogen bonding of your single-stranded RNA. Remember, this is single-stranded. But down at the bottom, you've got those three bases that make up the anticodon. Remember, anti means opposite or against. So we have the codon, which is the triplet in mRNA, and it matches up to the anticodon in tRNA. The same rules apply as when we were dealing with making mRNA during transcription. We're going to use a modified Chargaff's rule, A to U, C to G. Okay. On the right, you see a space-fill model, which is representing more of what the molecule actually looks like if you were to go ahead and visualize it inside the cell. Now, we've talked a lot about how the instructions are written in terms of DNA originally, but when we go ahead and do transcription, we're making a copy of them so that we can hand it out and not have the original blueprint leave the nucleus. But the ribosome needs to know where to start and where to stop making this sequence of amino acids. And the body has a way to go ahead and signal this to the actual ribosome. It always is going to begin with an AUG codon. Now, at this point in time, if you have your codon wheel near, I would really encourage you to go ahead and pull that out. If you go ahead and start in the center with A, out to U, out to G, what amino acid do you see there? That's right, methionine. Okay, right next to that, if you have a hard copy, I would go ahead and write the word start. You can use methionine in a lot of different places in a protein, but it will always be the first amino acid that we see in the sequence. There are, however, three different stop codons: UAA, UAG, and UGA. And the biggest thing I need you to remember here is that when you get to a stop codon, it does not code for an amino acid. This literally terminates the sequence. Okay? No amino acid is produced. That's the end of the chain. That's like the period of the sentence. You're done. Okay? So let's go ahead and give you an example. If we begin a sequence, you see AUG, that will produce methionine. You can go through and use your codon wheel, because remember, codon wheel is codons, so it's in mRNA, and that's going to tell you what amino acid it's eventually going to produce. You can go ACG, GUA, UUA, CCC, GAA, GGC, UAA. But when you get to UAA, it's just going to say stop on your codon wheel. This is because there is no amino acid that is produced. This is where it stops. This is where it terminates the sequence. Now, if you look at your codon wheel, you're also going to notice something else. Look for, I don't know, leucine, for example, or look for serine or arginine. When you go ahead and look at these more common amino acids, how many times does it show up on your codon wheel? Yeah, it shows up a fair amount. You're going to see 4, 5, 6 different combinations of bases, your codons, that are going to produce this particular amino acid. This is really important to understand because your body tries to be as simple as possible, but there are enough amino acids that 2 bases is just not enough for the codons. It won't give you enough combinations with the 4th base alphabet that we've introduced you to, A, U, C, G, to create all the 20 amino acids commonly found in proteins. Okay? It only produced 16. So we need more. But then you might be thinking to yourself, well, why don't we just go to 4? Well, then with 4, now you have more bases. The DNA molecule is going to be larger, which increases your risk for a mutation. So 3 ends up being the magical number here, where we have enough to produce all the different amino acids, but not so many that we're going to have a lot of generic nature to the genetic code and produce a lot higher risk of a mutation. If you look at the ones that are a little more generic, that have multiple copies, in many cases, they only differ by the very last base in the triplet. So if we take alanine, for example, we have GCU, GCC, GCA, GCG. So as long as the first 2 bases are G and C, it doesn't matter what the 3rd base is, it's still going to produce alanine. This gives you a little bit of protection against certain types of mutations. Okay? We're going to talk more about mutations when we get into next semester, but this kind of gives you a precursor thinking about why there might be this generic nature to certain points in the genetic code. So hopefully you've had a chance to go ahead and take a look at the codon wheel. Here it is again. Again, if you haven't figured out how to read it, let's go ahead and do a quick little review. You always begin on the inside. So let's go ahead and put a 1 here. Okay, you know that on the inside, this is your first step. Once you've determined which of the 4 bases is your first base, then you're going to move out to the second part, which is going to be this ring here. Okay, so whether you're going with G and going here, A and going here, C and going here, or U and going here, that's going to be your second round. So let's say we had UC. Okay, UC, this is going to be our 3rd round, and we could do this for every little trigger. I'm not going to write them all around here, but you see here that with serine, for example, UC, it doesn't matter what the last of base is here. Any of the 4 bases will go ahead and make this UC serine, okay, because it did start with the U, and that was followed by a C. So this little bit of generic nature to it is important. Now I have to realize that once again, when we go ahead and start taking a look at this, that when we talk about the codon wheel, what we're really talking about is mRNA, because codons are triplets in mRNA. Okay? All right. So if you remember, we finished transcription by leaving the nucleus through the nuclear pore. We have our nucleus right here. There's the nuclear pore from which the final mRNA transcript is going to be leaving. I say final because remember we had to remove the introns and keep the exons. When we go ahead and get to the ribosome, remember we have a triplet here in mRNA called a codon, which is going to match up to a triplet here in tRNA called an anticodon. And we're always going to begin our sequence with a particular sequence, AUG, which is going to give us methionine, the start codon. Once that's placed and the next one comes in, we're going to create a bond here between methionine and phenylalanine, and then we will release this tRNA to go pick up another methionine. Meanwhile, all of this is going to slide down, And the new tRNA comes in to match the next triplet or codon, bringing in the next amino acid, which will also form a peptide bond. Okay? And it keeps sliding down like this, adding one, adding one, adding one, until you get a long chain of amino acids. This is what's known as a polypeptide. Okay? This polypeptide chain will continue to be added to until you reach a stop codon. Now you'll notice when you hit a stop codon, there is no RNA, tRNA that matches with it. That's the way the body knows that this is the end of the sequence, and then it releases that polypeptide chain to go ahead and fold into what will become the protein. All right, so let's go over the entire process from start to finish now. We always begin where? That's right, in the nucleus. Why the nucleus? Exactly, because that's where the DNA is. We don't want the DNA to leave because we want to go ahead and protect it so that the original blueprint doesn't get damaged. Inside the DNA, step one is that we have to, good, unwind and unzip. Remember when we say unzip, we're breaking what kind of bonds? Right, hydrogen bonds, which hold together the bases. Then we have an important protein called RNA polymerase, which is going to copy the original strand of DNA to create our mRNA. Are we done? Hopefully you said no, because we still have to edit that initial transcript and cut out the introns, leaving only the exons to make the final mRNA transcript. Once this is done, then it's going to leave through the nuclear pore. Okay, mRNA leaves, DNA does not, and that heads out of the nucleus to the ribosome. At the ribosome, which remember has two subunits, a large and a small, they grab a hold of the mRNA, it assembles around it, and then it reads the mRNA triplets, which are called codons, and match up against the tRNA triplets called anticodons. That codes for a very specific amino acid. We begin with a start codon, which gives you what amino acid? That's right, methionine. And then it's going to begin bonding together more and more of these amino acids using what's called a peptide bond. It's going to continue doing this until it reaches a stop codon, which does not code for an amino acid. It just terminates the sequence and then releases the polypeptide chain that is produced, which folds into a protein. We have now covered all of the pieces of protein synthesis, starting with transcription, which takes place in the nucleus because that's where the DNA is. We don't want the DNA to be damaged, so we leave it in there, having the original blueprint protected. We copy the DNA into mRNA, but first we have to unwind and unzip it. When we unzip it, we are breaking the hydrogen bonds, and we're copying it using RNA polymerase, which is going to go ahead and copy the bases using a modified Chargaff's rule, A to U and C to G. And as it does so, it's going to end up with an initial transcript. This needs to be edited. We cut out the introns, keep the exons, which are reassembled to a final mRNA transcript that is then sent out of the nucleus through nuclear pore. That's transcription. Now, translation is when we go ahead and get that mRNA to where? That's right, the ribosome. The ribosome has two subunits, a large and a small, and they assemble around the mRNA. It's going to go ahead and read the mRNA in triplets also, but these triplets are called codons. It's going to begin at what's known as a start codon that codes for what amino acid? That's right, methionine. When it does this, it matches up its codon, triplet in mRNA, to the anticodon, triplet in tRNA, to give a very specific amino acid. It will continue to move in additional tRNA, creating a peptide bond between the amino acids, and then releasing the tRNA to get another amino acid. It does this for a long chain until it reaches a stop codon, which does not produce an amino acid and terminates the sequence. We have now produced a polypeptide chain, which is a whole bunch of amino acids strung together with peptide bonds, which will then fold to create our protein. Now, this last little part is more of a review just to go back what we talked about in terms of proteins so it's familiar with you inside your mind, okay? So you should already have this. This shouldn't be new. Remember, we have this traditional symbolic formula for an amino acid. On the left here, your NH2, this is what's known as an amine group, connected to a central carbon, which has a hydrogen on it, which is also connected to an R group. Now, the R group is the part of the molecule that actually changes from one amino acid to the other. And on the other side, you have your carboxyl group, COOH. In real life, however, it looks like this, more of a space model version, this one for cysteine. And you got to remember that all these models we're showing you are trying to help us visualize or understand what it looks like and how it reacts. But in real space, the space film model you see on your right is more accurate to what it actually looks like. There are a large variety of different amino acids. I won't belabor the point, but that R group is the only part that changes. NH2, we've got our amine group here, central carbon, carboxyl group. This is your R group. This particular one for cysteine is found using sulfur. Remember, if you remember CHOMPs, CHONPS, this is the only one that has sulfur to make disulfide bridges. You have some that are acidic over here. You have some that are basic, and it depends on the structure of the R group. But you always have the amine group, central carbon, and carboxyl group. Now, when it comes to proteins, remember there are so many different functions, everything from structural like collagen and keratin, regulatory, think hormones, right? Everything from insulin to adrenaline, human growth hormone, contractile proteins like myosin and actin, which help with our muscles, immunological antibodies such as gamma globulin to help fight infection. We've got transport molecules that act as carriers, things like hemoglobin, myoglobin, and we've got our catalytic proteins, those that help with our chemical reactions. These are our enzymes, amylase, lipase, lactase, so on and so forth. I don't want to spend a lot of time on it, and the last thing we need to know is protein structure. Remember, when you finish protein synthesis, we don't actually have a protein. We have a polypeptide, which is a sequence of a whole bunch of amino acids. If you remember when we were originally introducing you to proteins, we talked about how they have a quaternary structure, or at least a tertiary structure for your smaller proteins. These are going to be based on how they fold. Okay? So the primary structure of the protein is that polypeptide chain, the sequence of amino acids itself. That primary structure then is going to begin to fold into the secondary structure, which is those beta sheets and alpha helices, those secondary structures. Okay. Now, how those alpha helices and beta sheets fit together will determine the overall conformation or structure of the subunit. This is known as tertiary structure. And for your simpler proteins, this is going to be the entire structure. It won't get more complicated than that. But for most of our proteins, which are more complex and bigger than this, things like hemoglobin that you see here in your example, you have what's called quaternary structure. And in this case, you have multiple subunits. If you look at the example here, you've got an alpha chain here and another alpha chain down on the right. Those two alpha chains are both their own subunit, like you can see here in the example. And then there are two beta chains, one here and one here. So a total of 4 subunits make up this quaternary structure. Now, it may have 2 subunits or 3 subunits. It's not that 4 is a magic number, but you have more than just one subunit, which makes up the tertiary structure. And so that multiple subunits makes a quaternary structure. So I'll leave you guys today with amino acid abbreviations. You might record this down somewhere. It's just easier to write down the abbreviations, but not all of them are what you think they would be. If you take a look at isoleucine, for example, Instead of ISO for ISO, it's ILE, and these are the accepted abbreviations. And some of them could easily be confused with each other. Okay, so we think about like glutamine, for example. It's GLN because GLU was already used for glutamic acid. So this is a great way to use shorthand so you don't have to write out all the abbreviation, all the actual names of the amino acids. But if you want to do that, that's totally fine as well. All right, guys. Well, I hope you have a great day. I'll talk to you soon. Take care of yourselves. Bye-bye.